So good evening, everyone. Tonight we're gonna discuss uh, what is biotechnology. Okay, so you might wonder what is biotechnology in the presence of today's generation, science technology, as we say it. But before that, I'm gonna clarify uh, the discussion of biotechnology right after this. So we are a group, research group in biotechnology. Uh, my name is John Joshua Kalisanga. Next group is Aljun Guhai. And my third member of this group is Maria Loida Jordan. Okay, so let's proceed to this report. Oh, before that, I'm gonna present to you all the lists before we proceed to the topic. Okay, after this presentation, you will be able to know the meaning of biotechnology, history of biotechnology, types of biotechnology, use of biotechnology, importance of biotechnology, pros and cons of biotechnology. Right, so let's go, shall we? Um, History of biotechnology. In 1919, Hungarian agricultural engineer Carly Ericki foresaw a time when biology could be used for turning raw materials into useful products. So this example was emanated from the 1919 Hungarian agricultural engineer Karl Ericki when he first discovered that biology could be used for turning raw materials into useful product. So when we say raw materials useful product, that we are making products through using our resources such as cloth. Okay, cloth is made out of silk, weave, or even plants, even animals can make cloth. That's taken for example. And you might wonder that raw materials is very important to use for such variety of products. Like for example, this gadgets, laptop, cell phone, they all came from raw materials like such as treasures, like diamonds, or there's the screen, which is the glass, that was also a part of, I think that was what they call magnified and aluminum glass support. Some people might say that, but mostly it was used in laptop screens like this. So it came from a raw material as well. And also uh, things like things like um, local products, like here in the Philippines, we have native bags, wallet, basket, or such things like that, or even um, even like things to where you can put, or even box which is made out of wood, native products, or even sleepers. Okay, so those were the main examples of um, turning materials into useful product. So in Carl Ericke's time, he did make a research in biology when he first saw a time which perceived through his discovery that we can invent something which we can use in useful products, but by using technology. So that is the very start and the breakthrough of biotechnology use. So today we process uh, things and inventions through the use of technology, right? So that we can easily access them all. So it all started there in 1919 when Hungarian agriculture engineer Karl Ericke saw that. So when you say Hungarian agricultural engineer, which a person who is expert in agriculture, but uh, turning their dairy products or agricultural products into processing by the use of technology. So today we have rice, corn, crops like that, or even milk, which is now processed by full-time machine technological equipments to process them as a food manufacturing. Okay, so let's go to the next one. The breakthrough that laid the groundwork for modern biotechnology came when the structure of DNA was discovered in the early 1950s. Okay, so on this point, the groundwork for its source was found out in a DNA test example. Okay, so how did this one came out in biotechnology? Such as, let's say example, plant. Okay, a plant is very efficient and very scientific on its study purpose. So how can you define a plant by its growing or how the plant came to life, right? So a plant can be a seed or a little kind of a flower or leaf. That plant can grow up. So that structure was studied for a DNA sample. If that the changes, the dynamic of that plant will change into weeks or months or even years, like trees also. So everything is groundwork for DNA sample. Also animals, okay? So when we say animals, okay, I have this example because today in Singapore they use a manufacturing food processing machine, or the scientific the scientists in Singapore believe that food can animal foods like chicken or cow can grow up without slaughtering them. So what do is they get a cell from the animal, they process it into a lab. Once that cell will grow up, they will test it. Before that, they will test it in a DNA test if that animal is um, has a healthy background. If that animal was not sick or if that animal is still healthy. Okay, so they make sure that the food processing will be safety to all people to eat that kind of animal, like chicken or meat or pork or uh, beef. Okay, so they tested DNA, and once it grow up, they can already like it grow up for months. Okay, so it grow up for months because that shown in the video was a chicken cell which is used to be produced, and it will reproduce the cell to make food with it. So what do they do is once the food will be structured and manufactured, they do the task to make a food by baking it to turn it into a chicken nugget or fried chicken, okay, or even fried it. That was the main example that I have saw, or that only a DNA test can pass, okay, before turning that cell into a processing of food. That was the main example, but in this 1950 time, it wasn't discovered that time yet. But I surely believe that it was probably. Okay, so after that, in that time, one of the main examples that they used to use for experimental structure for DNA was like plants or animals in the early days, or because they have a structure of finding source of what scientific result will be affected. Let's say, for example, blood of an animal. Okay, so the blood of an animal can also pass into DNA tests that it can also help ground its structure for manufacturing products or resources. Okay, so one of the main examples is like I shouldn't say makeup, lipsticks that came from an elephant, like that kind of ingredient that they use. Okay, so that's it. Next is, what is biotechnology? Biotechnology is technology that utilizes biological systems, living organisms, or parts of these develop or create different products. Many forms of modern biotechnology rely on DNA technology, and it goes through biological process, such as fermentation and harness biocatalysts become manufacturing plants. Okay, so biotechnology is the modern biological system that perhaps it takes to work on detecting living organisms or parts of this to develop different products. So as what I said, so one of the main reasons that this happened is because uh, it changes through a lot in our modern society. But even back in the day, it was already used. Um, Biotechnology has a big impact in our uh, fermentation and manufacturing products. Not only plants, but also food, also medicines, also chemicals. So here we have the earliest form that manufactured as plants, because plants can easy to be test, or it uh, can easy to be used for manufacture. So when you say that plants came out from seed or like this or like that flower, or came from a soil, okay, so most people today who buy plants are made out of seed. So that's what they have. Uh, that's what they have to produce a product for the usage of manufacturing plants. Okay, so that is one of the main examples that most common in today in buying seeds of plants. Okay, so here are the two main topics of biotechnology, two main subject. First is fermentation. Fermentation is a metabolic process that produces chemical changes in organic substance through the action of enzymes. Okay, so fermentation is a process, metabolic process that produces chemical changes. Okay, so it is where the chemical change, the dynamic that which the substrate through the action of enzymes. So when we say that it is the substance that help changes the chemical reaction, like like take doing an experiment. There are a lot of chemicals to carry on and to be used, right? So the substance can change the dynamic through the ways of the enzymes. The action may change in a particular base of its uh, of its entailing experimentation. So let me say like organic experimentation of medicine. Okay, so a medicine can be also used for let's say for example that medicine is came from plants, and aside from that, excuse me. Okay, so aside from that, the 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 most particle of that experiment will change result. Okay, if a plant is made out of herbal, so the result will be a herbal capsule, herbal tablet. But before they do that, they experiment something like to test a result for the fermentation. The organic change, which the substance that carry for a use of chemical change reaction. So that is how it produces chemical changes, or experimenting chemicals that is dangerous or high risk. So let's say muriatic acid. It was the most dangerous acidic kind of chemical to be used in experiments. So once you put muriatic acid and you put a coloring, okay, let's say coloring, it has a chemical substance substrate reaction. The process of that something is they are collaborating with each other, but it changes also the effect of the acid and the coloring, liquid coloring. Okay, so that is where it happens that the process that produces inside the experiment well became organic and chemical change. Next is DNA technology. DNA technology is a sequencing, analysis, and cutting and pasting of DNA. This one is the analysis of cutting and passing DNA. When we say that it is to analyze a plant growth or its background, or humans to be used for their background. Or when we say DNA, it also relates to biological term. When we say biology, where it is where you came from, what are your status, what is your analysis, what is your detection, your existence. Also in organisms as well. such as plants, okay, like that, plants or animals. So they take DNA tests to know what this plant or animal background has or the scientific study of it. DNA is usually used today in hospital. I mean, in biological terms, like most people would actually ask who is the father of this child, who is the mother of this child. So the DNA will take that result, so that the biological term of the past. Okay, let's say the passing of that. Okay, the passing of DNA will get the result of the child support as who is his or her parents, maybe the father or the mother, will be tested in a DNA. But DNA technology, otherwise, it is also has the same sequence by using for products such as plants, foods, and animals, or like that. Okay, for knowing the background of this um, kind of thing, or what this thing is all about, what is its evidence and its historical background. Who is the father of biotechnology? Karoli Erecki, German. Karl Erecki, born on 20th October 1878, died on 17th of June 1952. He was a Hungarian agricultural engineer. He is regarded by some the father of biotechnology because he term biotechnology was coined by him in 1919. Okay, so Karol Erecki was a Hungarian agricultural engineer. He is regarded by some the father of biotechnology because he termed and coined the biotechnology in 1919. So when he found out the use of systems, because he was a Hungarian agricultural engineer. He was he was thinking about something on what to name on his on his profession. So he termed biotechnology in 1919, and it was coined by him. So he was the founder. He was the one who said that it is called biotechnology that we have in our research today. So if Karol Erecki wasn't there, or he didn't exist, the term biotechnology would not exist as well. Okay. The second is Louis Pasteur, 1882 1895. The French chemist and microbiologist is considered the actual father of biotechnology, the most renowned scientist of the world. It is he who had established the role of microorganisms in the fermentation process for the first time in the 19th century. Louis Pasteur is the father of biotechnology to the second, but with him, he discovered something different than Carlo Erecki, because Carlo Erecki was a agricultural engineer. Meanwhile, Louis Pasteur is a chemist. So when you say chemist, it's a chemistry. They experiment a lot of chemicals, bond, solving equations with the chemicals, the size or the quantity of that chemical to be used. The ingredients of that chemical is also tested for a result. So uh, Louis Pasteur established the role of microorganisms. So when we say microorganisms, it is a tiny in the little microorganisms. The fermentation process for the very first time in the 19th century. So it was that year when he was the renowned scientist of the world because of his popularity of establishing microorganisms. So maybe he experimented a microorganisms which is so little, and he tried to test it in a further way or detect it. As a result, it was an very spectacular experimentation by him. So let's just say that, because microorganisms can also be familiarized with. Micro means small, small, tiny little things, small particles. Okay, so that's it. Okay, so thank you for listening, and that is my part.